The overall goal of this procedure is to quantitatively measure the gene regulatory activities of cis-regulatory elements, or CREs, active during Drosophila melanogaster metamorphic development. Begin with site-specific integration methods to generate lines of CREs transgenically incorporated as reporter transgenes. Using a stereo microscope, obtain transgenic specimens of the correct developmental stage. Remove specimens from the outer puparium and mount them on a glass microscope slide. Proceed to record the level and pattern of fluorescent reporter protein expression in whole mount samples by confocal microscopic analysis. Finally, quantify the level of fluorescent reporter protein expression for recorded confocal images using the ImageJ software tool. These results assign a level of gene regulatory activity to a cis-regulatory element and facilitate further comparative gene expression studies on modified CRE versions.
This procedure can help answer key questions in fields interested in gene regulation and how that gene regulation is encoded in DNA, fields like developmental and evolutionary biology.
This procedure was developed to provide insights into cis-regulatory elements that function during Drosophila melanogaster metamorphic development. In principle, however, it can be applied to cis-regulatory elements that function during other developmental stages, in other fruit fly species, or even other model organisms.
In order to expand transgenic lines, set up 2 vials with several male and female flies. On alternating days, transfer the flies from one of the vials into a new vial. Repeat this for a week. By the end of 2 weeks, verify that the transgenic fruit flies range from larval to adult developmental stages. Start staging specimens at the end of the 3rd instar larval stage when the puparium is formed.